And I'm just going to warn you in advance that this session is going to be the longest one of the day and probably the most overwhelming one. It wouldn't be an OHDSI Symposium if there wasn't going to be something that's going to hurt your head just a little bit. And we usually ask Martijn to do that for us. And so Martijn's going to be delivering our plenary session here. We realized as we were preparing this that now that we've all gotten used to being on Teams meetings and all of our attention spans have drastically shortened, that nobody can listen to one person for 90 minutes straight. So you're going to be subjected to me just jumping in just so that halfway through this so that Martijn can get a sip of water and you can hear a different voice blathering on for a little bit. But make no mistake, this is all work that's been led and driven by Martijn that we're really excited to be able to share with you today. All right. I think we got most everybody in. So without further ado, I'm going to hand it over to Martijn Schuemie.
All right. Well, thank you, Patrick, for the introduction. And it sure is great to be here and meet everybody in person. So I'm very, very much enjoying seeing lots of people that I haven't seen before, only in Zoom or in Teams. So anyway, I will be talking about objective diagnostics, a pathway to provably reliable evidence, or as I later thought, maybe a better title would be how to avoid generating unreliable evidence. So I've actually been up here before, and I've talked to you before, and I've told you about that we actually have some issues with observational research. We have what you might call a reproducibility crisis, where many of these studies that are done, their findings are not being reproduced, even if we use the same data to answer the same questions. And overall, even though you might not say it if you talk to people in this room, overall there is actually quite a lack of trust in real-world evidence. And so we have to basically solve those problems. And I think the major issues why we have those problems are threefold. And we've outlined that in this publication that I'm showing you here. We have, first of all, observational study bias, which basically means that we're producing estimates that are incorrect because, for example, there is confounding or there's measurement error or well, other problems. And but another major issue is publication bias and p-hacking. And I want to illustrate that, those last 2 points with something I've done before, but I've redone it now just to reiterate the point that I made. So what I did before also was I wrote a small script that just runs through PubMed to extract effect size estimates. Like here's just a random abstract, and this script just pulls out these estimates. And so I ran this script for all the papers about observational studies. And so we wrote a PubMed query that identified 203,000 articles. And from those articles, we pulled 111,000 estimates, the way I just showed you before. So in this plot, I'm showing you those estimates. On the x-axis is the effect size, which can be the odds ratio or the hazard ratio or the relative risk or incidence rate ratio. And on the y-axis is the standard error, basically the width of your confidence interval. And so the higher you go, the wider your confidence interval is. And even though this plot is showing you estimates from all sorts of studies, different exposures, different outcomes, different methods, different metrics, I think you'll agree with me that there is a very obvious pattern in these estimates. And the cause of this pattern becomes very clear, I think, when you add these two lines. Because these two lines indicate where below those, the confidence interval no longer includes 1, where we basically reach, quote unquote, statistical significance. And in fact, if you look at this data set, only 17.4% of the estimates actually have a confidence interval that includes 1, and everything else is statistically significant. Now that number in itself might not be that big of a problem. It might be that researchers are just really good at finding research questions where there actually is an effect, and that they're actually powered to find that effect. Although I'm a bit suspicious of that too. But what really is a big problem in this plot is this sharp cutoff at p equals 0.05. Because that sharp cutoff can only be caused by two things, actually. Publication bias, which is the process whereby many studies that are done that have a non-significant finding never get published. Either they end up in a drawer, never get submitted, or if they're submitted to a journal, they never get accepted. And that actually leads to a hidden multiple testing problem that leads to massive false positives in the remaining set. Similarly, we see evidence of p-hacking. And p-hacking is usually done with the best of intentions. We have a researcher who believes something to be true. They run a study, p-value is greater than 0.05, and then they decide there must be something wrong with my study. Let me go back. They find something that they want to tweak, and then they keep on doing that until they find the significant finding that they were expecting. That actually also invalidates your finding. Again, leads to hidden multiple testing and to many false positives. So as the end result is that this set of findings that you have in literature actually is highly unreliable. And so in OHDSI, in the past years, we proposed a way to tackle these problems, all three of these problems, in LEGEND. LEGEND stands for Large-Scale Evidence Generation and Evaluation in a Network of Databases. And in LEGEND, we follow 10 principles that are aimed to solve these three problems of observational study bias, publication bias, and p-hacking. So these principles are described in the paper that I'm showing you here, and these are those 10 principles. I'm not going to read all of them to you, because that would be a little bit boring, but let me highlight the most important ones. So principles 1 and 2 state that we should generate evidence at large scale, so not just for one exposure, one outcome, but for many exposures and many outcomes in the same study. And we should disseminate the evidence from such a LEGEND study, not looking at any of the results to decide whether or not we'll disseminate them. So everything gets disseminated, whether they're statistically significant or not. And of course, those two rules, where you combine them, they effectively eliminate publication bias. The next two principles state that the evidence will be generated using a pre-specified analysis design, consistently applying a systematic approach. So that's not to say that we believe that there is a one-size-fits-all for all of these questions, rather that the way that you modify your approach for every question is something that in itself can be turned into a systematic process that you can then apply. And if you pre-specify that, that means that you can't actually change things afterwards, after you see the results. So that effectively deals with p-hacking. The next two principles are that we should use best practices for dealing, for example, with confounding, and that we will use control questions. So let me highlight those two a little bit more. So for currently, our best practice, which might change in the future, is that we will use large-scale propensity scores to adjust for confounding. So LSPS, what we do is we construct a very large set of generic covariates, so they're not specific to the exposure or the outcome, and that typically is somewhere in the range of 10,000 to 100,000. So basically one covariate for every concept in the common data model. We then use a regularized regression to fit our propensity model, and then we match or stratify on the propensity score. And interestingly enough, what we see quite, well, in many cases, is that if we apply this approach, then after the matching or stratification, we end up balancing on all the covariates that we observe. So here on the x-axis is the standardized difference of the mean before matching, and on the y-axis is after matching. And we see that these two groups that were very different to begin with now are very similar on all the covariates that we measure. And if they're similar, then that means that there can be no confounding from the things that we measure. So this approach has been described in this paper a while back, but I would like to highlight this paper that was very recently published, led by Lin Ying, where Lin Ying actually goes into more of the theoretical foundations of why large-scale propensity scores work, and why you need to have to worry about things like colliders, and why this approach actually can't really adjust for completely unmeasured confounders, but it can adjust for confounders that are only indirectly measured. So this theoretical basis, I think, is a very strong foundation for us to keep using large-scale propensity scores. The next thing that we typically do in OHDSI studies, and especially in LEGEND, is we measure residual systematic error using control questions. And so the idea is very similar to when you have a scale and you want to know whether that scale is accurate. Well, what do? You put something on the scale where you know the weight, and then you see if the scale actually indicates the weight that you know it has. And so similarly, in OHDSI and especially in LEGEND, we use control questions, which are exposure outcome pairs, where we know what the true effect size is. And then we run our study and we see whether or not it actually produces the estimate that we believe to be correct. And so we typically use negative controls, where we believe that the null hypothesis is true. And so, for example, the hazard ratio is 1. And we also tend to generate positive controls, but those tend to be synthetic. Once we've ran those control questions, we can actually see how much deviation there is, and we could actually, just like you can calibrate a scale, we can calibrate our statistics, such as the p-values and confidence intervals, to adjust for any of the systematic error that was observed using those controls. And those approaches have been described in these papers. So the last couple of years, we actually had several studies that I would characterize as LEGEND studies. The first one was on depression treatments. It wasn't officially called LEGEND at the time, but we did follow many of the LEGEND principles there, and we actually initially developed them within that study. Then there was the first official study, the LEGEND hypertension study, and we talked at length actually at this symposium about this study, where we looked at all hypertension treatments, compared them for a large set of outcomes. The SCILA study that I'll talk about a little bit in a minute also followed LEGEND principles, and currently the next LEGEND study is underway. This is a study on estimating effects of diabetes treatments, type 2 diabetes treatments. And so we recently published the protocol for this study, and this work was led by Rohan and Mark. But I'll mostly be talking today about the LEGEND hypertension study and its results. That study already resulted in several high-impact papers, including in The Lancet, Hypertension, and JAMA. And as George already mentioned before, there actually seems to have been real impact already in clinical practice from these papers. So like I said, LEGEND looked at all hypertension treatments, both monotherapy and combo therapy, and so we had lots of estimates that we produced in this study. I'm here plotting all of them, almost 8 million of them, which is a lot. And so the same paradigm as before, effect size, in this case hazard ratio on the x-axis, standard error on the y-axis. What you probably notice is that this distribution is very different from the one that we saw in literature, because obviously there is no publication bias or p-hacking going on here. And that, I think, in itself, I think we underestimate how big of an impact that has. That in itself means that this set of results that we have from the LEGEND study is much more reliable than what's currently in literature, even if we look at the subset of literature that is specifically on hypertension treatments. So each of these 8 million estimates that we produced in LEGEND hypertension comes with a set of diagnostics. For example, we provide information on the statistical power, the covariate balance, and the systematic error as observed using these negative controls that we used in LEGEND. Now the question is, how do you then use these diagnostics? So in LEGEND, we just provided them for whoever wants to use the evidence to interpret. And if we look at, for example, the book of OHDSI, it also mentions diagnostics and says, on the basis of these diagnostic results, a decision can then be made whether or not to move forward with executing the final outcome model. But that feels a little bit soft, right? How exactly should we make this decision? And who makes this decision, and on what grounds? So that's actually the main story that I want to talk about today. So diagnostics, study diagnostics, other diagnostics, inform us on whether we can trust the results of a study. And I've actually been here on this very podium, although it was moved a little bit They're arguing that you shouldn't unblind your results unless you pass all diagnostics. And I've actually shown you this diagram before. It's actually also in the book of Odyssey, where we argue that you shouldn't finalize your protocol until you have all the diagnostics generated and passing. And only then do you register the protocol and execute the full study and look at the results. We did not follow that specific paradigm in LEGEND. Instead, we chose to generate all the evidence and provide the diagnostics for you to then apply those considerations afterwards. For example, if you look at the papers that we published, there the researchers that wrote the paper had to check all these diagnostics before including results into the paper. So since then, we also had the SCILA project. So SCILA was a LEGEND-like study that looked at the safety and effectiveness of drugs that were proposed to treat COVID-19. In this study, we had 650 treatment comparisons, 31 outcomes of interest, and several different analyses per comparison and outcome of interest. And this was work that was presented last year by Mark Suchard at the OHDSI Symposium, the virtual one. And Cheryl Tan has also presented this at the recent ICPE meeting. Now, SCILA used a slightly different approach to diagnostics. It moved basically ahead from LEGEND hypertension in that it had predefined rules for some of these diagnostics, like equipoise, covariate balance, and statistical power. And in SCILA, we only unblinded results after we verified that they passed these 3 diagnostics. What was a very interesting and very sobering finding in SCILA was that actually only a small fraction of the estimates that we produced passed the diagnostics, somewhere between 1.5 and 8.9%, depending on which database you looked at. That is a very low number. So there are challenges with study diagnostics. Currently, the interpretation of the diagnostics is mostly subjective. I would argue if you fail a diagnostic, you should stop the study. But that is actually a big ask. If you've invested a lot of time and energy in the study and then you should stop and not do anything else, that might be a bit of a disappointment. I've been there myself. I've worked on studies where I invested weeks to get the study up and running, and then I produced the diagnostics, and then we looked at it and said, nope, we can't do this. We should not move forward. I do feel good about that, because else we would have generated unreliable evidence. It's even a bigger ask if you already looked at the results. Luckily, I didn't do that, so I could just say, we're not going to actually generate the results if we're not going to trust the evidence. Also, a big issue that I realize is that failing a diagnostic is currently not publishable. So you did all this hard work, you came to the right conclusion, but then there's no paper that's going to publish this, no journal that's going to publish this. And also what we saw in SCILA especially is that, so we were very diligent and looked at these diagnostics and didn't look at the results if they failed diagnostics, but there were many other researchers out there who were looking at the same questions, using the same methods, but didn't look at diagnostics, and they still published the results that we at least discarded because we thought they were not going to be reliable enough. So diagnostics are hard. Are they worth it? Patrick.
Yes. So at the start of the year, at our first Odyssey community call, we talked about what we wanted to accomplish together as a community, and I tried to provide an argument for what I thought was wrong in the current field and where I thought we could possibly go here in 2022. And one of the things I argued was that the current status quo of observational research makes it really challenging for us to build trust in evidence. We've got this process right now where some people write protocols, others don't. There may or may not be some sort of review process for a protocol, but basically once you have an idea of what you're going to go do, you go through this process of figuring out the right data you're going to use, extracting out your cohorts, conducting some sort of analysis, looking at the evidence, and you choose to disseminate it. Oftentimes we disseminate it by drafting a paper that's submitted for peer review. And it's only at that moment that the reviewer, reading the draft paper, has to ask the question, is the evidence reliable? And that review is highly biased by the results themselves. If the results conform to what they believe, they may be more likely to believe that study is credible, and if the results defy what they expect, then they may be more skeptical. But ultimately, that question about is evidence reliable isn't about the results, it's actually about the rigor of the study. And there's real concerns that happen at the very end of this process that makes you look back to figure out what's going on. There's methodologic concerns about bias and measurement error and data quality. And there are also technical concerns about, like, is the programming done correctly? And is the logic for phenotypes correct? And how is the data standardized? And so my assertion at the start of the year was that we have to figure out a totally different paradigm for building trust. And what I posited as an idea at the start of the year that I challenged several of you to start working on was to think about how we could actually engineer an open science system that systematically builds trust in the evidence generation and dissemination process. And I showed this picture at the start of the year where I made the argument that what we need to do is we need to start with a pre-specified system specification that has all of the required phenotypes, all of the analysis specifications, and all of the decision thresholds of how you're going to determine what information is going to be deemed reliable. And that needs to be decided up front. And once we have those decisions up front, we can walk through a stage gate process whereby first we assess data quality and determine which databases are fit for use. And only those databases that pass those database diagnostics move forward, and those other databases stop. The second stage is to assess phenotype development and evaluation, whereby we run cohort diagnostics to determine which phenotypes are fit for use, and they can move forward. And when we learn that a phenotype is not appropriate, we stop. We then move forward to analysis reliability evaluation, and we talk about study diagnostics, as Martijn has alluded to. And only after we pass those study diagnostics do we move forward. Otherwise, we stop. And by having these stops along the way, what it means you're left with is only the information that you should actually be unblinding. And at that point, that is the honest information that we can trust, and we can freely provide that information through some sort of interface for exploration. So this is the idea at the start of the year that I threw out to the community, and I said, I think we should start working on this. And I'm super happy to report a lot of the progress we've made towards this progress. So the first I'll highlight is database diagnostics. The basic idea that I would assert is that database selection right now is a really subjective and opportunistic process. We don't actually start by saying, what's the universe of all data available and which ones are best? We're usually asking the question like, what's in hand, or is the data that I've got going to be acceptable? And what I would assert is that we need to have objective criteria with pre-specified decision thresholds that let us figure out which databases across a network are actually suitable for our purpose. And even more ideally, across our OHDSI network, if we had aggregate summary statistics about each of those 450 databases that have now been converted to the OMOP Common Data Model, if we had aggregate information, we could actually figure out which databases across our whole network would be appropriate for a study. Well, Claire Blackadder's taken this on and developed a really exciting tool that has been released this year called DB Diagnostics. It's out on the OHDSI GitHub, and you should take a look at it. And effectively what it does is uses the Achilles aggregate summary results and provides analytics on top of that for you to specify your research question and determine whether or not your database is going to pass the first stage review. And just to highlight what that output could look like, what I'm showing you here is output for one of the questions in the LEGEND hypertension study, asking the question, which databases are fit for use for a comparison of lisinopril versus benazepril for the outcome of acute myocardial infarction? Every row here is a database. Every column is one of these database diagnostics. If you see green, that means we've passed the diagnostic. If you see red, that means we've failed the diagnostic. So we can see across this sample of databases that Claire run this analysis for that some of the databases are failing specifically because the data source doesn't capture inpatient visits. And in our phenotype for acute myocardial infarction, we required that those events happen in a hospital. So that means these databases are not going to be fit for use for that particular question. Now, some databases fail because they don't actually have sufficient longitudinal follow-up. If we're going to do a study of hypertension patients and on treatment time and then look in the future for an MI, that means we have to have some period of time of continuous observation, and a hospital-only database like Premier isn't going to cut it. Now, some databases aren't going to pass the test because they don't have the comparator drug. CPRD and IQVIA Australia don't have benazepril, and so therefore they're out. And other databases may just not have sufficient exposure. The JMDC database in this case only had a few hundred estimated patients to be able to study, so that's not appropriate. But what we're left with is 6 databases that pass all of our database diagnostics and are estimated to have adequate sample, so we can move them on through the process. Now, what I'm excited about, what Claire has done in this work, is that it used to be at J&J, we had access to this set of databases, and my team, I love them all, but they would usually go, oh, I'm going to use MarketScan today because I'm familiar with MarketScan, or this study I'm going to use Optum because I like Optum. Choice of database isn't about a preference. Choice of database is about figuring out what data are fit for use. And now we have an objective tool that can actually help us determine which databases are appropriate for which questions, at least as it relates to the first stage. So I'm really excited about the work that Claire's done, and I hope that you talk to her and help her advance this moving forward as we think about how to build this out across our entire network. Second stage, phenotype diagnostics. So the challenge we have is that we have to create phenotypes to identify exposures and outcomes, but all of them are subject to measurement error. And the challenge is that right now we don't have objective criteria with pre-specified decision thresholds to determine whether or not a phenotype is actually adequate. And so what we need to be doing is developing standardized processes for developing the phenotypes, but also being able to estimate sensitivity, specificity, positive predictive value, index date misspecification. And if we can quantify measurement error, we can actually incorporate that directly into our assessment of whether or not the phenotype is fit for use. Now, this year, I'm really proud of our community for making lots of progress in the space of phenotyping. George mentioned about Phenotype February, and I encourage you to check out Azza Schwebie's lightning talk later, where she's going to talk about some of the lessons learned there. We've also seen a lot of maturity in our community in developing open source tools to support phenotype development and evaluation. Jamie Gilbert has been releasing the cohort diagnostic tool. Martin Lavalle's released CAP-R this year. And Gautam Rao's released the Phenotype Libraries, all open community resources that are now available to mature this process. Joel Swerdell published a paper on Phevaluator 2.0, which actually provides us a probabilistic model for estimating measurement error. And he's got 2 really cool posters next to that. I'd encourage you to go check out. But we're starting to get the sense that we can actually estimate measurement error. And at the Odyssey Europe meeting last year, Jamie Weaver presented about how we can actually incorporate measurement error into our effect estimates. And actually, we identified that some traditional approaches for quantitative bias analysis probably are not actually reasonable for use in a lot of the types of questions that we're interested in. And this weekend, Jamie and Gautam and Azza are leading a workshop specifically to try to get into how do we determine that we've got an acceptable phenotype. So I encourage you all to join that. But what we're going to focus on for the rest of this plenary is study diagnostics. And the challenge here is that when you make a choice as a researcher to design an analysis, you risk producing a misleading estimate because of the choices you make in your design. And so if we could have objective criteria with pre-specified decision thresholds that can allow us to determine whether or not our analysis is acceptable in regards to precision, accuracy, and generalizability, then we can actually have greater confidence in our results. Now, I'm going to assert that this idea of study diagnostics applies to any type of analysis you could do on a data set, whether that be a characterization study, a population level effect estimation study for safety surveillance or comparative effectiveness, or whether you're applying machine learning algorithms for patient level prediction. In this plenary, we're going to focus on causal inference and population level estimation, but I want you to listen to the messages of what we're saying and think about how that could apply to any study that you're doing. If you are trying to generate evidence from a database, I would assert study diagnostics is something that should be directly applicable to you. And while we're going to focus on causal inference, I encourage you to go check out Jenna Repp's poster, because she's actually got a poster that specifically talks about how diagnostics can and should be applied to patient level prediction. And in fact, she's already implemented in the Shiny app, so all you got to do is just run her code and you get it. So it's just great work from Jenna on the PLP side. So what we're going to focus on is stage 3, analysis reliability evaluation. And we're going to more specifically focus on the comparative cohort design, because that's the method that we applied in the LEGEND hypertension study. And so we're going to focus on that to try to demonstrate how we can actually establish these objective thresholds. But I want you to be thinking more broadly just about, if we're going to start with a network of databases, if we can pre-specify objective rules, have the discipline to stop when we fail a rule, then the end of that process will be evidence that we can trust. Now, Martijn's going to go through a crazy amount of results in just a couple minutes. And so I'm going to just try to introduce what these 5 study diagnostics could be for a comparative cohort design, just to give you an intuition of what the ideas are so that you can try to follow along as Martijn goes through the evidence base. So the first is the idea of statistical power. And the metric that we will be using is minimum detectable relative risk. Now, statistical power is an idea of a hypothesis test. Basically, it's the probability of detecting an effect if a real effect exists. Oftentimes, statisticians will talk about type 2 error as the metric. And very often when we talk about power, people think about prospective data collection, whether it be a randomized clinical trial or somewhere where you just do primary data collection for a registry or whatnot. And there, what you really are doing is you're doing power calculations to figure out how much data do I need to collect. But we're doing retrospective analysis of existing data. The data is already there. So instead, you can frame the power question around and say, given the available data, what's the effect size that this analysis is able to detect? And we could all probably agree that more data is better because it will give us better power, greater power, so greater precision. But it turns out the analysis choices you make will influence how much data actually comes into your process. And so a potential diagnostic that we could consider is how much data is sufficient to provide useful information. Now, I think we could all agree that if you were given the choice between more data and less data, all else being equal, more data is better. That should probably be clear. What's actually a little bit less clear is if you had a choice between less data and no data, what would you choose? And the challenge is when you are in the situation of less data, there's a risk that you are going to misinterpret statistics, particularly underpowered statistics that give you crazy wide confidence intervals. The number of times people misinterpret a point estimate or confidence interval is kind of nauseating. And so this debate, is less data or no data better, is an open question in the broader research community. This year, Miguel Hernan published a commentary in the Journal of Clinical Epidemiology called Causal Analysis of Existing Databases, No Power Calculations Required. And in this commentary, Miguel actually basically made the argument that, listen, the goal is effect estimation, the goal is not hypothesis testing, and I don't really care how precise your estimate is, just give me something, because as long as we can synthesize the evidence later, we're good. That was not universally received well. Miguel's commentary resulted in multiple letters to the editor from various esteemed statisticians saying, hold your horses here. There's more that's going on than just producing this effect estimate. But one of the core arguments that was made from these rebuttals to Miguel's commentary was the assertion that, listen, we know that Miguel would never screw up interpreting statistics, but other people will see a crazy large point estimate with an extraordinarily wide confidence interval and misinterpret it. And we could do harm if that information gets out there. Miguel actually had to write a response to the response of his commentary where he said, well, at least we're finally talking about doing causal inference and observational data, and we can kind of agree to disagree on everything else. The point here is that this is not a settled issue, but statistical power could be something we could specify a rule to decide on. Let me just show you quick good examples and bad examples from LEGEND hypertension just to give you a gist of this. One of the studies in LEGEND hypertension was a comparison of lisinopril versus hydrochlorothiazide for the outcome of cough. And in the LEGEND app, if you were to go out there, you can see the minimum detectable relative risk. And you can see that there were 7 databases with a minimum detectable relative risk of less than 1.75 and 5 with a relative MDRR less than 1.1. That is to say, we were powered to observe an effect size of 10%. Most people wouldn't even trust an observational study that had an effect size of only 10%, but at least we had adequate data to be able to reveal an effect that small, given the prevalence of the exposure and the outcome. But not all studies in LEGEND were like this. If we looked at candesartan versus chlorthalidone for the outcome of rhabdomyolysis, we saw we only had 3 databases, and their minimum detectable relative risks were greater than 6, and actually 2 of them were greater than 15. Not only that, you can see that that estimate was based on one database having less than 5 cases and 2 databases having 0 cases. So the question really is legitimately remains. Should we look at an effect estimate from this study? Because there's some information here, but that very, very scant information might actually be harmful. Changing gears, study diagnostic number 2. The idea is empirical equipoise. The metric is the preference score. Now this is a pre-adjustment design diagnostic asking the question, what proportion of your target population is close to treatment indifference? To draw an analogy to a randomized clinical trial, when we randomize in a head-to-head study, we flip a coin, it's a 50% chance that someone gets treatment or placebo. If we could design an observational study where the treatment choice was approximately 50%, we'd be pretty happy. But of course, treatment choices in a non-interventional study are decided by the patients and the providers. And so we use the propensity score model as a way to assign a probability of treatment to these individuals. But if we could find the people that are really close to 50% preference, that would be like finding the people that are close to a coin flip. So this idea of a preference score was introduced in this paper by Alec Walker several years back. It actually hasn't gotten very many citations, even though I think it's a really, really nice paper I'd encourage you guys to read. In this paper, Alec and colleagues described a tool for assessing the feasibility of comparative effectiveness research. And they introduced this basically rule of thumb that they walk through, which says like, take a drug, take a target, take a comparator, fit a propensity score, normalize it to the proportion of target and comparator that you had, and then accept it as a viable comparison if, step 5, accept the drug pairs as emerging from equipoise if at least half of the dispensings in each of the drugs are to patients with a preference score of 0.3 to 0.7. So here they've specified a rule that one could follow to make a choice of whether the comparison's reasonable. Now, there's no empirical basis for that number 50%. There's no empirical basis for why it's 0.3 and 0.7. But nonetheless, Alec provided a nice rule of thumb that could be followed. And he provides some mathematical justification for how you can calculate the preference score that I encourage you to take a look at. Now, this idea of equipoise is something that we actually think could actually be a hardened rule to follow. If we look in LEGEND, we can see good comparisons, like comparing 2 different ARBs, valsartan versus olmesartan. If we look at the preference score on the x-axis and the distributions of those values, we can actually see that in this particular study, greater than 90% of the people are near empirical equipoise, meaning that they have a preference score between 0.3 and 0.9. So that's to suggest that a person is largely indifferent between their choice of valsartan versus olmesartan. So we should feel good about a comparison there. But that's not like every comparison. If I take valsartan, but I swap out the comparator for chlorthalidone, you see the distributions of the propensity scores look like this. Here, we only have less than 30% of the patients near equipoise. And that is to suggest that the baseline characteristics of people who choose valsartan versus those who choose chlorthalidone are fundamentally different. And one has to ask the question, if there's such a preference for one treatment or the other, is this actually a valid comparison to make at all? So we could have a rule that could actually help us understand how close to equipoise do we need to be to feel comfortable. Diagnostic number 3, covariate balance. The metric is standardized mean difference. The basic idea is that we are worried about confounding. The relationship between exposure and outcome could have some third confounding effect. And one way to mitigate confounding is to make sure that there is no imbalance in exposure for any possible measured confounders. So this is a post-analysis analytics diagnostic where we can ask the question, are all observed baseline characteristics sufficiently similar between the target and the comparator cohorts? And of course, we've got various different strategies for trying to do adjustment to try to make this appropriate. We've been using this in the OHDSI community for a while. There's tons of papers by Peter Austin talking about balanced diagnostics, but when you read into them, you realize that all of Peter's papers basically say the same thing, which is there is no clear consensus on the issue of what the threshold should be, but some researchers have proposed a standardized difference of 0.1 as denoting meaningful imbalance in the baseline covariates. So Peter's got like, I don't know, 10 papers saying that sentence, that same citation, going to a paper from Sharon Leese Norman. And in her paper, she talks about how she's using the Cohen effect size, which is basically just another name in behavioral sciences for the standardized mean difference. And she asserts that a small, e.g. less than 10% absolute value, supports the assumption of balance, and she makes some citation here. If you actually follow that back down, you actually get all the way back to Jacob Cohen's textbook, where he says reluctantly, I don't really want to give you rules of thumb to follow, but since you want them, I'm going to give you a small, a medium, and a large, and he defines small as 20%. So everyone is reticent to actually set a hard rule about this, but nonetheless, these rules kind of become ingrained in our culture. And actually, Martijn and I have been up here, and we've seen posters where everyone says, we use the Austin rule of 0.1. Austin ain't saying that he asserted it was 0.1. He's saying there's no clear consensus. But what if we could achieve consensus? What if we actually could define a rule that actually had some sort of empirical basis? Now, we've got examples of good studies. Amlodipine versus atenolol. You can see we had 45,000 baseline covariates, but after adjustment, all of them have a standardized mean difference of less than 0.03. We've got bad studies. Candesartan versus atenolol, when we applied propensity score stratification, we had 50,000 baseline covariates. All of those blue dots above 0.1 are variables that did not achieve balance. Not only do you see that there were variables before adjustment that were out of whack, but we can actually see that the propensity score itself induced bias by actually expanding the difference in some baseline covariates. So we could have an objective pre-specified rule to decide, is this good enough or should we stop? Fourth metric, generalizability. And the metric, again, is going to be a standardized mean difference. Here, generalizability is just the extent to which a study result can be applied to some target population that you're interested in. This is another post-adjustment and analytic diagnostic. And we're asking the question, are all observed baseline characteristics sufficiently similar between the pre-adjustment target and the post-adjustment analytic cohorts? So imagine you've got this target cohort of a random set of people, each with their own characteristics. If you were to select a very large collection of that as your analytic cohort, then what you'd probably find out is the baseline characteristics between your target and your analytic cohort are probably going to be relatively similar, even if the selection of those was non-random. However, if I were to select a relatively small subset of our target cohort within my analysis group, I might find that there are now systematic differences between who I was left with and who I started with. And those systematic differences could mean that I am going to produce a valid causal inference, but my causal inference doesn't actually relate to the population that I actually want to talk about. And this question of generalizability is something we need to pay attention to. There's nice papers talking about various metrics for standardized mean difference. This paper by Tipton et al. I encourage you to take a look at, but it suggests standardized mean difference is one of them. And it also says, researchers have borrowed rules of thumbs generated in observational studies and have used rules such as 0.25 or 0.1, and there's Sharon Leese again popping up with her cardiovascular paper. Good study, lisinopril versus losartan. We started with 650,000 patients. We ended with 647,000 patients. That's 99% of the people remaining. Standardized difference, very small. Happy about that. Bad study, lisinopril versus atenolol. We start with 650,000. We end with 85,000, meaning only 13% of the exposures are left. And we can see that the standardized mean difference, there are variables that are making these groups systematically different. Should we stop? Number 5, residual bias. The metric is expected absolute systematic error. And the idea here is a post-adjustment analysis diagnostic. Is the residual bias observed from negative controls small enough to accept that a calibrated effect estimate can really be trusted? So imagine you've got a scale at home in your bathroom and it's reading 126.4. Now, many of us have scales at home. They may be off by a pound or two in one direction or the other. And you kind of know how to just handle that. It's directionally correct. You get on it and you're like, okay, I know how to kind of correct that. But if I put a 100-pound weight on a scale and it read 126.4, you've got a choice to make. Are you always going to subtract 26 pounds from your weight, or are you going to buy yourself a new scale? And that is basically the question about how much calibration is too much calibration. Now, Martijn mentioned we published several papers on this, and the metric is just the quantification of the empirical null distribution. We've got examples in Legend of good studies. George's paper, hydrochlorothiazide versus chlorthalidone, showed that there was very little residual bias, and therefore calibration had almost no effect. And we can actually see the effect estimate didn't even move. But we've got bad studies, furosemide versus lepidolol, where the effect estimate starts at 5.5 and calibration moves it all the way to 2.8. Are we going to trust the fact that we had to make that magnitude of a calibration? And then there's studies that are kind of in between. So furosemide versus chlorthalidone, we can see the effect estimate moves from 1.6 to 1.2 upon calibration. And the question is that too much? Is that too much to be credible? So what I'm going to assert is that what we need, what we started at the start of the year and what we're making great progress on, is we need to come up with rules that we can objectively follow, that we can pre-specify at each stage of this process. And we're making good progress on database diagnostics. We're continuing to make some progress in phenotype, and we need to continue to grow there. But for the analysis work, the question is, how do we produce an empirical evidence base to understand what these thresholds should be and how much we can trust them? And I believe that diagnostics can provide evidence that can build trust, but as Martijn said, post hoc interpretation is just going to lead to investigator bias. And the current thresholds, as I hope I've convinced you, is like they're all just random rules of thumb with no evidence base behind them. If we can actually develop empirical evidence to set these thresholds, I think we're going to be in a much firmer foundation to be able to actually trust the evidence that we've got. So Martijn's going to go through some really exciting work we've done to actually show you how we can use the LEGEND results to figure out exactly what these thresholds should be.
All right. Well, I'm painfully aware that I'm the guy standing between you and lunch, but I'll continue my story anyway. So how can we actually tell whether these diagnostics are having an impact? Well, why don't we use the LEGEND results that we produced as a way to do that? So you can all go to the LEGEND evidence viewer and, for example, here, look at one of these estimates here. We can plot that estimate in a similar plot as I did before, and we can just plot all the evidence that came from LEGEND in this plot. Here I'm restricting to just the 471,000 estimates that were for monotherapy comparisons and only using the on-treatment time at risk, and also excluding meta-analysis. I did that just to make it a little bit more manageable, because plotting 8 million dots can be a bit of a pain. So this is the distribution that we see of estimates. And we're probably going to see that distribution move as we apply our diagnostics. But can we say something about how good this distribution is? Not really, because we don't know what the true effect sizes are. So these could all be spot on exactly where they need to be, but we don't know what the ground truth is. So maybe we can define a subset of these estimates where we actually don't expect there to be an effect. So one of the things that's going for us is that these hypertension medications have been around for a long time and have been well studied. And we can look at the product labels, which tend to be very inclusive for adverse reactions. So if a product is even remotely believed to might cause an outcome, it tends to be on the label, so it's highly sensitive. And so if we combine those two things, then we can come to a conservative approach in defining such a subset. For the list of outcomes in LEGEND, when comparing two drugs, if neither the target drug nor the comparator drug has the outcome on the label, and going even further, if even none of the other products in the same classes as the drugs has the outcome on the label, then we're pretty confident that both drugs don't cause the outcome, and therefore the hazard ratio is probably around 1. So an example, if we look at ACE inhibitors like lisinopril, well, they are known to potentially cause angioedema, so that's very clearly on the label. Calcium channel blockers and ARBs are not really believed in general to have this side effect, but still it's listed in the post-marketing experience, so that rules them out from our set. However, none of the direct vasodilators and alpha-1 blockers have angioedema on their label. And therefore, for example, if we look at hydralazine, which is a vasodilator, and we compare it to prazosin, which is an alpha blocker for the outcome of angioedema, that's actually pretty likely to be null. Similarly, ARBs like losartan have rhabdomyolysis on the label, but beta blockers and loop diuretics do not. So if we look at metoprolol, which is a beta blocker, and compare it to furosemide, which is a loop diuretic for rhabdo, then that is likely a null finding. So using that logic, we constructed a set of 9,752 target comparator outcomes. That's actually symmetrical, so actually you could argue it's just half of that. And this now forms a new set of imperfect negative controls, imperfect in that we're not 100% sure that the null is true for all of these. Now you might say at this point, well, wait a minute, Martijn, we already had a set of negative controls in LEGEND that we used to calibrate, for example. What's the difference between this new set and this old set? Well, this new set we will not use just within a specific comparison in a specific database. We will use it actually across all of the analysis. So we actually, hopefully, will have a little bit more power, even though for every single comparison, we actually have fewer controls. The outcomes are more similar to the outcomes of interest than in the original set of negative controls. And that is because these outcomes of interest are actually things that are known to be caused by some of the other drugs in this LEGEND study. And so maybe that will give them a better exchangeability in terms of more similar confounding to the outcomes of the questions of interest. I don't know if this matters, but also these outcomes have full phenotypes behind them with complicated logic, whereas for the original set of negative controls, we simply used any occurrence of the concept or any of its descendants to find the outcomes, the negative control outcomes. So here I'm plotting just that subset of estimates that came from LEGEND, where we believe the null is likely true. Now this distribution we can look at a little bit more closely. So these are just 11,716 of the calibrated estimates from LEGEND. That's slightly bigger than the 9,000 I mentioned before, but that's because we have multiple databases for the same question. If we look below this dashed line, so these are the estimates that have a confidence interval that doesn't include 1. And we here see that there are actually 15.2% of the estimates fall into that category. If the null is truly true for all of these, which is probably true, then you would expect only 5% of these to have a confidence interval that doesn't include 1. And so in this case, 15% is quite a bit higher than 5%. A more accurate way to look at this deviation from the null is by looking at the fitted null distribution that I'm showing you at the top. So we had a mean and standard deviation estimates, but we can even summarize those two numbers in the metric called ease, or expected absolute systematic error. The intuition there is if this ease score is 0, you can only observe that if two things are true. Only if the null is exactly true for all of the estimates, and if there is no error other than the random error expressed in the confidence intervals of these estimates. So here we see an ease that's 0.38, which is actually quite high. So we currently don't know exactly what it is, but we do have the intuition that if we propose a diagnostic rule and it brings down this score, then we're actually moving in the right direction. Another way to look at the fitted null distribution that I like to look at is this visualization of these orange areas, which is if you were to do empirical calibration where after calibration things become significant. We're not doing calibration with this new set, but I just like to show these areas, because if these areas exactly coincide with the dashed line, that means again that the ease will be 0. All right, so let's use this now as a tool to look at the effect of diagnostic rules. Patrick talked about the statistical power expressed as minimum detectable relative risk, and let's set a threshold that it should be smaller than 10. Reasoning that even low-powered estimates could be helpful, but if the power is really too low, then probably that would be misleading. I should note that in LEGEND hypertension, we already required exposure cohorts to have at least 2,500 subjects, so that probably already eliminated many of the low-powered estimates. So I'm showing you here a histogram of the distribution of the minimum detectable relative risk in that set of 470,000 estimates. And we see that many of them actually had a very low minimum detectable relative risk, only 5% failing our 10 rule of 10. Patrick's example of a good study is all the way to the right. His example of a bad study was even still here in the green area. It should be noted that most of the effects for antihypertensives have a hazard ratio smaller than 0.2, so this cutoff at 10 isn't really not about determining whether we have enough power. In fact, we do. It really is to get rid of these hard-to-interpret estimates. So like if you get a hazard ratio of 5.1 with a confidence interval from 0.7 to 36, like lots of people get hung up on the 5.1, even though the estimate isn't really telling you anything because the confidence interval is that wide. So I'm here showing you the effect of that rule on the distribution of estimates. So these are all the 471,000 estimates. On the left is before applying the rule, on the right is after applying the rule. And as we already saw, we just lose 5% of the estimates, and that doesn't seem to have a really big effect. If we now restrict to the subset where we believe the null to be likely true, then we also don't see a lot of change in this distribution. And actually, the E-score is still exactly the same. Probably that's because we already guaranteed quite a lot of power with that rule of having at least 2,500. So let's go to the next diagnostic that Patrick mentioned, which is equipoise. So Alec Walker proposed this threshold that equipoise should be greater than 0.5, which basically means that at least 50% of your population in your study has a preference score between 0.3 and 0.7. The reasoning is that if the equipoise is low, then the populations are really just too different, and we might not trust our ability to make them comparable. So if we look at the distribution of equipoise scores in this set that we're looking at, actually quite a few of them have low equipoise. 71% fail this rule that Alec proposed. Patrick's example of a good study where the preference score distribution greatly overlapped is all the way to the right. His example of a bad study where they were very much separated is over there to the left in the red area. As you might expect, this distribution looks very different whether you're looking at comparison within a class versus between a class. So if comparing within a class, then equipoise tends to be high, and between classes, there is a very high probability that your equipoise is low. So if we now filter by that rule, then we see that we lose a lot of estimates, but we also see a marked change in the distribution of these dots, right? We don't really know if that's a good thing, so that's why we also can look at this subset where the null is likely true. And so here we see something very interesting. After applying this rule, the ease is almost zero. And as I mentioned before, the only reason you can see that is when the null is true for everything and if there is no systematic error left. And so this plot actually demonstrates these two things at the same time, that our set of likely null is almost certainly null, but also that applying this rule, even though it loses a lot of estimates, actually greatly improves the quality of this evidence. Now, we don't like to lose this many estimates, so maybe we can make this rule a little less strict. So we looked at relaxing the rule to just 0.1 instead of 0.5. If we look at the overall distribution, there's not much changing here. And if we look at the set of likely or almost certainly null, then we do see some change in the E-score, in the amount of systematic error remaining, but not as big as when we use the threshold of 0.5. So maybe this in itself isn't such a good idea. Covariate balance. So we've been using this rule for a long time. that it should be, the maximum standardized difference of the mean should be no greater than 0.1, which, as Patrick showed, actually is a pretty arbitrary rule that has no real foundation. The reasoning here, if the covariates are unbalanced, then there may be confounding. If you look at the distribution of this maximum standardized difference of mean, we see that actually there are quite a few studies also failing this rule, 56%. Patrick's example of a good study is all the way to the right. Patrick's example of a bad study with lots of imbalanced covariates falls to the left of this threshold that we've set. What is very interesting to note here is that actually, depending on whether you do propensity score matching or stratification, you're much more likely to pass this rule. If you're doing matching, it's very likely that you pass the rule, and if you're doing stratification, it's much more likely that you don't pass the rule. We looked into this a little bit. The reason for that is if you have a small group of people who are really fundamentally different, then if you do matching, you just basically throw them out of your analysis. But if you do stratification, you still have to squeeze them into one of these strata, and therefore you end up basically still having an imbalanced population. If we filter by covariate balance, we lose a lot of estimates, but I think we do see some change in this overall distribution. And if we focus on the ones where the null is likely true, we also do see a shift in ease from 0.38 to 0.28. So there is a reduction here in systematic error, but if I'm being honest, it's not as much as I would have expected. So equipoise had a big effect, but equipoise is something you compute before propensity score adjustment. And I don't know why that had such a big effect, but this is something that we compute after we've done a propensity score matching or stratification. I would have thought that would have directly informed of how much confounding and systematic error is remaining in the study, and actually it's not as big of a deal as I would have expected. Patrick also talked about generalizability. So this is comparing the target cohort to the analytic cohort. So the target cohort is the ones that you identify in your database that have the exposure of interest. And then the analytic cohort is a cohort that's left after you, for example, did propensity score matching and eliminated people that had the outcome prior. And if those are very different, then you might not be able to generalize from the one cohort to the other. We use maximum standardized difference of mean now between those two cohorts, and we set a rule that that has to be smaller than 0.25. We see again quite a few of the estimates failing this diagnostic, 57%. Patrick's example of a good study to the right and a bad study to the left of this threshold. Again, we see a big difference between whether we are doing propensity score matching or stratification, but this time in the opposite direction of what we saw before. So now here we see that if we do propensity score matching, then we're very likely to fail generalizability, where if we do stratification, we're very likely to pass generalizability. Whereas balance, we saw that for matching, we were failing the rule very often, or passing the rule very often, and stratification, we were failing the rule very often. That's because these are, of course, the flip side of the same thing. If you're adjusting your population a lot, then afterwards you hopefully have balance, but now you've adjusted so much that you've moved away from the population that you started with. Filtering by generalizability, I don't know, it doesn't seem to have an impact on the overall, a big impact on the overall distribution. If you look at the likely null set, there also is not really a big effect. And maybe we shouldn't expect that actually, because this generalizability rule isn't so much about systematic error, it's about generalizability, so maybe not that surprised. What is very interesting, though, is if you combine those two rules, the balance rule and the generalizability rule, all of a sudden there is a big effect. And that's because there is an interaction between those two. So if we require both balance, which I showed doesn't have that big of an effect, and the generalizability rule, which I showed has virtually no effect, then all of a sudden together they have a very big effect. Hard to explain, but just to point out that actually there are interactions between all these diagnostics. The final diagnostics is systematic error expressed as the expected absolute systematic error, or EASE score, where we set a threshold at 0.25, where the reasoning is that even though we can and should empirically calibrate for the residual error that's measured using the original set of negative controls, we might not trust it if we have to calibrate too much. So note that our evaluation is actually looking at the calibrated estimates, so that's already incorporating basically the ease in the calibration. Luckily, there are not that many estimates that are failing this diagnostics, only 16%. Patrick's example of a good study where there was hardly any effect of the calibration is all the way to the right. Patrick's example of a bad study is all the way to the left. If we look at the effect on the overall distribution, it's not that big. If we look at the set of likely null estimates, then there's also not that big of an effect, I would say. My interpretation is that it doesn't really have a big effect because the empirical calibration was already basically moving away all of these estimates that had a really big ease score. So we have now 5 diagnostics. And on the bottom left, I'm showing you again, just to remind you, all the thresholds that we picked for every one of those diagnostics. And then we can see, well, how many of our estimates fail none of these, 1, 2, 3, 4, or 5. And so that's what's shown in that histogram. And you see that only 12% actually passes all 5 of these diagnostics. And in fact, more than half pass at least, or fail at least 2 diagnostics. If we look at the effect of passing all of the diagnostics, that's actually a substantial effect on this distribution. It's much narrower than it was before. And if we look at what this effect is on this new set of negative controls, this likely null set, it's also a profound impact. After applying all 5 of these diagnostics, we get an ease score that's rounded to 0.00. Meaning again that the null is likely true for all, but also that there is no systematic error remaining, whereas in the original set there apparently was quite a lot of systematic error left. So applying these rules does have a big impact on the reliability of the evidence that we generate. I should remind you that in LEGEND, like it's not that we produced bad evidence in LEGEND, we just required you to apply these rules. Like when we wrote these papers, we also had to look at the diagnostics before we wrote those papers. Can we do better than this, actually? Because yes, we reduced the systematic error to 0, but we lost a lot of estimates, and we would like to generate more evidence instead of less. These current thresholds are arbitrary. What if we just treat this as an optimization problem? Because we just have 5 thresholds that we can move about. And we have an objective optimization criterion. We want to maximize the number of estimates that remain after applying the rules, and we want to keep the residual systematic error very low, lower than an e-score lower than 0.05. So that's what we did. We just iterated over all possible combinations of these of the values for these 5 thresholds, and tried to see what is the optimal setting. And so here the middle column is showing you the thresholds that we had before that were derived from literature. And on the right-hand side, you see the new optimal thresholds. It is interesting to see that this optimization proposed that we don't have any threshold on statistical power, generalizability, and systematic error. And that sort of confirms what we already saw, and that they didn't have much of an impact on systematic error. So why even have a threshold there? But I'll get back to that in a second. It does propose keeping Alec Walker's threshold of 0.5 on equipoise, which unfortunately is a very strict rule, and that the covariate balance rule threshold can be moved all the way up to 0.5. So for the first time, we actually have a non-arbitrary estimate of what would be a good threshold for standardized difference of mean in terms of covariate balance. If we apply these rules, instead of keeping 12% of our estimates, we can now keep 29% of our estimates. So that's a lot more. If we use this set, like I said, we keep 29%, we fail 71%. Most of them actually fail just 1 diagnostic. That again has a big impact on the overall distribution. And if we focus on the set where it's likely null, then basically by design, the E score is very low, so we lose a lot of systematic error. And we get to keep more estimates than our original set of rules. Now, some of these diagnostics actually weren't intended to remove systematic error. The MDRR threshold was for improving interpretability, and The generalizability, well, that was just for improving generalizability. So we might not expect them to actually impact systematic error, but if we care about them, we should also pre-specify them. So let's say I add a new constraint to my optimization, which is just that, for example, I want generalizability to have a threshold of 0.25. So now all the way to the right is showing you under that constraint, including the constraint that I want systematic error to be really low, what is then the optimum? And now all of a sudden we see an interesting phenomenon that the equipoise threshold can be lowered to 0.25. Covariate balance has to become stricter, has to become 0.15. And if we apply that set of rules, then we keep 23%. So let me wrap this up. We've shown that diagnostics can help improve the reliability of evidence as measured by systematic error. Other diagnostics might have different goals. Up to now, diagnostics rules were arbitrary, and our empirical evaluation provides some evidence for choices of these thresholds under the various constraints that you might set when you start a study. Now, one thing that we haven't talked about a lot is that these diagnostics rules probably need to pre-specify it. If you allow post hoc interpretation of these rules by the investigator after they've seen the results, they might actually, that might lead to bias. Because if I get an estimate that I really like, but then I fail a diagnostic, I might say, oh yeah, but I got the answer that I think is right, so let's ignore the diagnostic. So the rules should be pre-specified, for example, in the protocol. And so Patrick showed you already this plot where there are all these stop signs where you should stop when you fail a diagnostic. What it just showed you is that you should expect to stop a lot. So that is a big aha, I think, for the Odyssey community, is that we'll probably be needing to stop studies a lot more than we've done in the past. So you need to evaluate these diagnostics before looking at the study results. That can be either by including the actual diagnostic results in the protocol before you continue with the study, or like we did in LEGEND, or we should do in LEGEND, you pre-specify your diagnostics rule in your protocol. The really important thing here is just not look at any of the results that don't pass diagnostics. So I think this is all part of an evolution that we've seen in Odyssey. So we had originally our traditional observational study where defining the research question, generating evidence, interpreting the study reliability, and then interpreting the results was all ad hoc and expert-driven, where expert-driven with me usually has a bit of a negative connotation in that it was subjective. In LEGEND, and especially LEGEND hypertension, we tried to make those first two steps pre-specified and using a systematic approach. So both defining the research question, so instead of hand-picking a research question, we just looked at all questions we could think of for hypertension treatments. And then the evidence generation, as I talked about earlier, was a completely pre-specified and systematic process. But still, we left it up to the consumers of the evidence to interpret study reliability. And so what we're proposing here is to now go the next step in the evolution, where we make this interpretation of study reliability by looking at these diagnostics and by pre-specifying their thresholds, turning that into a pre-specified and systematic approach. I left one box orange, so let's talk about that for a little while. If we look at, for example, results from many databases, here one of the LEGEND results, what do we do with that? Do we just look at the meta-analytic estimates and ignore all of the evidence from the individual databases? I don't know if that's the optimal thing to do. If we look at evidence from different analysis, for example, stratification and matching, and they differ, what do we do in that case? Do we ignore one of them that we don't like, or is there a way to synthesize across those? And so I hope that the next evolution in Odyssey will be that we also create a systematic and pre-specified approach for this interpretation of results. One thing that I briefly wanted to talk to you about, and then you can go to lunch, is another way of using these results. And that's what we call discovery. So here is this distribution of estimates that I showed you before, where we applied our new optimal rule set of diagnostics. And so we know that this evidence set probably is very reliable. We've eliminated publication bias, p-hacking, and observational study bias. So this is a really good set, I would argue. If you see this plot, like me, are you then not itching to look at these dots? What are these? There are effects there. Some of them pretty large. Are they known? If they're not known, that might actually be very important, because if we looked at those, we might actually impact patient lives for the better. Well, we've never done that before, and the reason why we've never done that is because now we get into a tricky area where we start to have to adjust for multiple testing. So the way that we've currently argued you should use the LEGEND results is similar to how you use literature, in that, say a doctor has a question, what is the effect of ACEs on acute MI compared to ARBs? The doctor goes to this set of evidence and then pulls out one of these estimates. What the doctor might not have said, but might have been thinking is, I want to use an alpha of 0.05, meaning a type 1 error of 5%, so 5% probability of getting a false positive if the null is true. In this case, the doctor had one hypothesis. She looked at one result, so that's one test that she needs to correct for to get the desired alpha, which means no correction. So that confidence interval is actually the right one for her to use if she wants to maintain an alpha of 0.05. Just as a side note, but I do find it very interesting, if that doctor had instead gone to the literature, as I showed you, because of all the rampant publication bias and p-hacking, she would actually have to adjust for all the hidden multiple testing going on there, but she can't because she doesn't know how much is going on there. So actually this evidence base of LEGEND is, I would argue, much more reliable than the evidence that we currently have in literature. But now we're proposing a different scenario of discovery. What if there's a researcher who is asking the question, what effects achieve statistical significance? And out comes a list of effects. Again, this researcher would need to specify some operating characteristic. For example, that the family-wise alpha is 0.05. And what it means is that there's a 5% probability of having at most one false positive in this list if the null is true for all. To answer that question, you actually need to consider all the results. And now, to achieve that desired alpha, you actually have to adjust for all of the estimates that you're looking at. In this case, 136,000. So that's going to be a heavy adjustment that we'll need to make. Interestingly, and I haven't thought about this very much, but because we fail so many diagnostics, we have fewer tests to adjust for. So actually, we are gaining power by removing estimates. I'm not sure how that will balance out in the end. But anyway, so we want to use this approach in effect surveillance. And I think one of the goals that we should set as the OHDSI community for ourselves is to build a surveillance system for monitoring multiple treatments, for multiple outcomes across multiple times at risk, using multiple methods, both comparative effect methods, or maybe also self-control designs, multiple databases, and probably looking multiple times across calendar time. So as a new product comes on the market, we maybe look at it after a few months, and then a few months later we look again. All of those are multiple tests that we need to adjust for. And the question that we still have to answer is how best to do that. Not only that, I'm not even sure what the best operating characteristics are that we want for such a system. It's definitely not a family-wise alpha of 0.05, because I don't think that will ever be the optimal thing to do for patients. But then what is the correct thing to strive for? Luckily, Van Boe has already done some initial research in this area, so I very much recommend that you check out our talk at 1 p.m. here on this very podium. So, wrapping it up, then you can go to lunch. So diagnostics are important to ensure reliability of results. And Patrick and I have been saying that to you for quite a while now. I hope that this is the first time that we actually show some empirical evidence to support that, and that you'll finally believe us. I do think that OHDSI needs to become more rigorous in applying these diagnostics. We should turn this interpretation of these diagnostics into a systematic process, and we should either evaluate these diagnostics before even starting the study, or pre-specified diagnostics rules. The most important thing being that you just don't look at results that don't pass diagnostics. We should also be aware that many studies will fail diagnostics. And I think that actually is a good thing. I think less evidence is better if the evidence that remains is evidence that we can really trust. It would be good if we could disseminate the failures to pass these diagnostics. And I think this actually argues why we should do more legend-like studies, because there the failures to pass diagnostics actually become part of the result set. Looking forward, there are many opportunities to propose and improve these diagnostics, so there's still work for us to do. I would like a better balance metric, because I think the results that we saw was that we didn't have a good balance metric, maybe better equipoise metric or generalizability metrics. We need to have good go, no-go rules for data diagnostics and cohort diagnostics. And as I alluded to, we need to start thinking about how to improve the interpretation of results, how to synthesize evidence from multiple databases and multiple analysis, and most importantly, how do we design a discovery system or a surveillance system, and what are actually the operating characteristics that we want for such a system. And with that, thank you for your attention. And lunch is ready.